The next presenter is Sandra Thibivilliers from the University of Nebraska-Lincoln from Mark Libo's lab. Sandra?
Well, thank you, Gergo, for the invitation. And I would like to start actually by saying that I was very excited about hearing the first talk because it overlaps a lot with what I will show. And it's very interesting to see that different people with different background actually came out with the same conclusion. And it's very validating because it shows that we are going on the good direction, or I don't know, we are all making the same mistakes maybe, but I really don't want to think about that. So today I will show the progress that we made into exploring the single nuclei multiome analysis. I joined Dr. Libo lab about 5 years ago. And the position was really about, was really about developing a single multiome approach that could be broadly applied to a lot of different plant species and more especially to crops. And so 2 people already presented similar information. And so we decided to focus on nuclei because we could use it to perform RNA-seq, ATAC-seq, and multiome. And they are more easily accessible. And also, because we access only the polyRNA available in the nuclei, they are better correlated with the epigenome. By saying this, although it means that we have less RNA accessible into these nuclei, and so we have to be very careful about RNA leakage. So the first step into establishing a multiome approach was to develop some strong protocol to isolate nuclei compatible with that approach. And we started with developing a protocol to isolate nuclei compatible for single nuclei RNA-seq. And so the protocol has been published in 2020, and we are still using it in the lab. And so very briefly, we harvest our sample and we chop it into a nuclei isolation buffer. After incubation, we filter out the biggest debris and we perform DNA staining in order to do fans. So I don't think I have to go over it. It was very well described earlier. Once we collect our purified fraction of nuclei, we perform a centrifugation. We took the side of eliminating the buffer that is used during the fan in order to not interfere with the downstream reaction. We centrifuge them, resuspend them in a PBS complemented with BSA and RNA inhibitor. And after cell counting, we perform some single nuclei RNA libraries using the 10x genomics. As you can see here, you have a Medicago root sample that Dr. Cervantes published back in December. And we isolated the nuclei from this root, and we were able to recover about 1,300 expressed genes per nucleus. More recently, we apply the same strategy to perform some single nuclei RNA-seq isolation, but in this case, using the scSeq technology. And so the beginning of the isolation of the nuclei was very similar. We chop our sample, incubate it into a nuclei isolation buffer, filter out the biggest debris. But in this case, we actually did not perform any fans because the requirement in the number of nuclei in order to perform this library was a little bit too high. And so we moved directly from filtering to centrifugation. And we resuspended our nuclei directly into the buffers that are used by parse to fix the sample. Upon fixation, we can keep these nuclei for a few weeks to couple months at the -80, which is very helpful when you have a large amount of sample to process. So when you are ready, you can move on to the construction of your library using the scSeq approach. So we use a Parse Bioscience kit, and we were able to generate some libraries. Here is an example of samples that we process. Again, it's some medical root in this case. And using this approach, we were able to recover 1,900 express genes per nucleus. So we were pretty happy with this approach, and we were also pretty confident that the protocol we are using is pretty robust. In the lab, we're working on crops, and one of the crops of interest is sorghum. And we perform actually the analysis of single RNA-seq library on sorghum root, and we apply both technology. We apply 10x and Parse, and we compare them. And so, as you can see on this UMAP, the library generated with the 10x technology and the Parse Bioscience technology were integrated, and we can see that they're completely overlap. We had 2 replicates for the 10x, 1 for the Parse. We recover 1,900 express genes with the 10x technology and 1,400 with the Parse. But overall, they could give us exactly the same information, and we couldn't see any bias into using one or the other technology when analyzing the data. So we are pretty confident about this protocol, and we were able to generate some good single nuclei RNA-seq. And so the next step was also to develop a protocol to access the gene accessibility in these nuclei in order to perform some multiome analysis. And so the protocol started about the same way. We isolate our sample, chop it, filter out the big debris. And here came the question about performing fans or not. And we asked the question because most of the fluorescent DNA staining will interfere with the chromatin organization. A DNA staining will leave some kind of fingerprint on your DNA, and this fingerprint can actually interfere with the TN5 activity and create a bias in the ATAC fragment that you will recover. So here is an example of the interference of the DNA staining on the ATAC fragment generation. So we used a root, isolated the nuclei, we stained them with propidium iodide, performed some fans, isolated some good-looking nuclei. We perform an ATAC-seq library. As you can see on the profile here, generated with a bioanalyzer, we have a good profile that actually looked pretty exciting. But after sequencing and alignment of this fragment, we generated a UMAP that was very globular and for which the identity of the different nuclei was not very clear. And we lost definition into this cluster. And so, like Egon said earlier, we reached out to 10x and asked them for help. And they directed us to a web page where they actually explain that several staining will interfere with the TN5 activity, and that if you use them, you will lose identity of your nuclei and you will not be able to recover the information you were seeking. And so there were only one staining that was recommended, which is the 7AD presented earlier. The only inconvenience with 7AD is that the emission spectrum is the same window as the chlorophyll. So it could work very well with the root, but not really with green tissue. Consequently, we opt out of performing FAMs when generating some single nuclei ATAC-seq library. So upon centrifugation of our nuclei, we resuspended them into the nuclei permeabilization buffer. We use a pretty harsh treatment of triton, which was at 0.5%, because we were not really concerned about losing RNA from the nuclei, as we were not targeting them in this specific analysis. And so upon cleaning and washing these nuclei, because we wanted to remove any trace of triton, as triton denaturate protein, and we didn't want it to actually denaturate the TN5. So upon washing, we resuspend our nuclei into an ATAC buffer and perform some single nuclei libraries. And upon sequencing alignment, we were able to generate some UMAP for which we had a better definition of our clustering. So as Egon presented earlier, the goal of this approach was to look for the integration of the single sNucRNA-seq and the single ATAC-seq, and to see if we could draw any correlation. And yes, indeed, we can draw correlation between these two data sets. And so we could access the impact of the gene accessibility into the regulation of the gene transcription. And if we look at it a bit closer, we can see here, so on the top you have a bulk ATAC-seq. On below it you have the single nuclei ATAC-seq data, and you can see that all the major peaks detected in bulk were also detected with our single ATAC-seq. But more importantly, we could detect more discrete fragments that were present only in a few clusters, here cluster 14 and 15. And these peaks are actually in the transcription start site of one gene here. And when we look at the expression of this gene in our single nuclei RNA-seq data, we can see that this gene is very specially expressed in the cluster 14. And so we can draw some conclusion about the importance of open chromatin on this site for the expression of this gene. So this approach rely on the acquisition of the transcriptome and epigenome, but it's done on two different fractions of nuclei. And so as described by Egon, you now have access to a kit that allow you to access the transcriptome and the epigenome from the same nucleus. And so we decided to go back to the protocol and optimize it in order to use this kit and access this information from a same entity. We started with the same step. We collected our sample. So far, we only work with fresh sample. We chop it, incubate it to release the nuclei, and filter it out with a two-step of filtering. Based on our experience with the ATAC-seq, we of course opt out for doing any fan, and we move directly into the resuspension of our nuclei after centrifugation into the nuclei permeabilization buffer. And as it was said earlier, the concentration of the detergent, but also the detergent themselves are very important. Different detergents will actually affect different parts of your membrane. They can target protein or cholesterol. And so it's very important to choose them properly, but also to choose their concentration adequately. As said earlier, if you over-permeabilize your nuclei, you can damage them to the point where you cannot recover any information for them. And again, if you over-permeabilize them, you can also have a leakage of RNA that will make your nuclei very sticky, and you will have the formation of aggregate and clump. And so when we start to see this aggregate, it means that we went too far into the permeabilization of our nuclei, and we start losing the RNA or any other information, which means that we have to take a step back and we have to revise the composition and the duration of the permeabilization of these nuclei. Once we reach a good composition of detergent with a good timing, We can move on to cleaning these nuclei, removing this detergent, resuspending our nuclei into the attack buffer to perform the tagmentation. And it's very important to not over-permeabilize your nuclei because they will stay for 1 hour at 37 degrees. And so you don't want them to leak during this step. Once you finish your library, I'm almost done, sorry. You can get this kind of output. So here you have a multiome made of soybean root where we recover about 7,000 nuclei, for which we have on average 12,000 unique peak and a little bit over 1,000 gene. And when you combine these two sets of information, this is a kind of map that you can generate. So you have the transcriptome and the epigenome integrated in this one. Here you have a single sNucRNA-seq, also based on the isolation of nuclei from root, and we were able to isolate 1,300 genes per nucleus, which is not so far from the 1,000 coming from the multiome. So we are fairly confident that we were able to recover nuclei at good quality and will give us some good information. Overall, we were able to recover a good amount of gene and attack fragments that link to each other, which is very exciting. And here is a quick example of the quality of this information. So we use a Resolve Bioscience to perform some MERFISH, and here you have a probe that targets the phloem. When we look at the gene expression of this probe, it's very specific to this cluster, the green one, and so we can say that this cluster is most likely the phloem one. And when we look at the ATAC-seq side, we can see that this gene is linked to some fragment that are present only in one cluster, the phloem cluster. So we are fairly happy with this data and we are currently analyzing them. So I would like to thank all the people who participate to this project, Dr. Xu and Ben Smien, who helped with the generation of the libraries, Dr. Cervantes and Andrew Farmer for the bioinformatic analysis, and Sutton Tennant, who generated the bioscience, the Resolve Bioscience picture. Thank you.
Thank you, Sandra. It was really interesting to hear all these techniques applied for plants.